It is my honor and great privilege to introduce our speaker today, Dr. Robert S. Langer, the David H. Koch Institute Professor at the Massachusetts Institute of Technology. I had the privilege of joining Bob's lab about 20 years ago as a graduate student, and he continues to inspire me, to act as a mentor to me, and to promote me in every respect. When I met Bob, it was about 10 years after the artificial heart had been implanted here at the University of Utah, and I was very interested in pursuing a degree and research in medical device research and development. And I met with Bob, and he said, let's not worry about making artificial hearts out of metal and plastic and parts that could fall apart or degrade or go away or fail over time, but he said, we're going to grow hearts. And that was very inspiring, that we could think about using the biology of cells and the extracellular matrix that they create to create new organs. Bob has been a pioneer in the field of tissue engineering and regenerative medicine, in addition to his work in drug delivery, pharmaceuticals, and in medical devices in general. He has received many awards that are listed in your program, all of the top awards in engineering and science. He has published close to 1,200 papers and has 800 patents. So he's one of the most innovative and prolific writers that's living today. He also has a family. He has a wife and 3 children that are all developing, and he's very proud of them. He spends a lot of time with his students, mentoring them and advising them, and he's very proud also of the many students that have gone on to accomplish great things in industry and in academia. He really is a great friend and a great individual. Please help me in welcoming Dr. Robert Langer to the stage.
Thank you so much. Thank you. Well, thank you very much. It's an honor to be here today. And I thought I'd tell you of some of our work in biomaterials in the areas of drug delivery and, at the end, tissue engineering. But before I do that, and as a way of starting, you know, a lot of times people ask me, well, how does somebody like a chemical engineer get into the medical field? And I thought I'd tell you back to when I was around your age, sort of some of the decisions I went through and how I thought about this. So I got my doctorate at MIT in 1974. In 1974, just like a few years ago, there was this gas shortage, and the prices of gas kept going up and up. But then it was even worse. At least in Boston, if you had a car, you had to wait in line for over 2 hours to get your gas tank filled. But the consequence of that was that if you were a young chemical engineer, you got a lot of job offers. And pretty much every one of my classmates at MIT in 1974 went to an oil company. And I thought that's what I should do too. So I interviewed at the oil companies. In fact, I did pretty well. I got actually 20 job offers, four from Exxon alone. And it wasn't like I was that great or anything. But one of them made quite an impression on me. I remember going to Exxon at Baton Rouge, Louisiana, and when I was there, one of the engineers said to me, if I could just increase the yield of this one chemical by 0.1%, he said, that would be wonderful. He said, it'd be worth billions of dollars. And I remember flying back to Boston that night thinking to myself that I really don't want to do that. And I started thinking about what I did want to do other than work in an oil company. And one of the things I thought about was education. You know, I'd helped start a school for poor kids in Cambridge. And when I was doing that, one of the things I did was develop new chemistry curriculum. And this was for poor kids, poor high school kids. And one day I saw an ad in a chemistry journal to be an assistant professor at City College of New York to do just that, develop chemistry curriculum. So I wrote them a letter applying for this job. But they didn't write me back. But I really liked the idea, so I found all the ads I could to do that at other schools. I found about 40 of them. I wrote 40 letters applying for assistant professor of chemistry education, and actually none of them wrote me back. So that wasn't going so well. So I started thinking, are there other ways that I can use my chemical engineering background to help people? And I thought of health. So I wrote to a number of hospitals and medical schools, and they didn't write back either. But I thought, you know, so that wasn't going so well. But then one day, one of the postdocs in the lab where I was at said to me, he said, Bob, he said, there's a surgeon in Boston named Judah Folkman. And he said, sometimes he hires unusual people. He thought very highly of Dr. Folkman. I won't say what he thought about me. I'm going to start by showing you a picture in the New York Times in 1971 of how Dr. Folkman thought about trying to treat cancer. This was a radically different idea, but let me start with this. So his idea was that a tumor somehow became abnormal. The cells became abnormal. It's actually a chemical engineering problem, he said. It grows to no larger than 1 millimeter cubed, or 1 million cells, because the cells run into a nutrition problem. Cells in the center can't get nutrition and can't get rid of waste. They die. Cells on the outside can proliferate, but you get this equilibrium. Can't get to be more than 1 million cells, except for the fact that he said tumors make a substance called TAF, tumor angiogenesis factor. And what they do is that causes the surrounding blood vessels, which normally do nothing, to start growing directionally right to the tumor. And it feeds it. It stops the nutrition problem. And so the tumor now can grow, get bigger and bigger because it gets all that nutrition, and actually can spread through those blood vessels to other parts of the body. That's a process called metastasis. And that's how a lot of tumors kill. Dr. Folkman's idea, which was very radical at the time, was that if you could stop blood vessels, you could stop cancer. And so when I came there, of course, I just thought this sounded like a great idea. I didn't realize it turned out that almost everybody in the biology field thought this was totally wrong. But when I went there, I thought it was very exciting, and he offered me a job, which turned out to be a job to discover the first angiogenesis inhibitor, the first substance to stop it. So how do you even think about a problem like that? Well, what I thought about was, well, there's a substance in our body that doesn't have blood vessels. It's cartilage. So I decided, let's see if we can find something in cartilage that can stop it. So what we did is I found a slaughterhouse, and I got a cow bone, and I took the cartilage. But I couldn't get that much that way, so I found out where do all the cow bones go from this slaughterhouse, and actually from every slaughterhouse in the greater Boston area. They went to a meat packing place in South Boston. I made an arrangement with them to get all their bones, and I brought them back to the hospital. And here's one of them. And I spent about 50 hours a week scraping meat off these to make it look like this. I'd slice this off, and I put it through various extraction and purification procedures. So I'd have about 50 different vials of substances I wanted to test. That then leads to the second problem. How do you even think about testing something to stop blood vessels? What kind of bioassay could you use? And actually, if you look throughout the history of medicine, penicillin, lots of things, this is usually what halts progress, is not having a bioassay. So how do we think about it? We wanted something at least semi-quantitative. We thought, you know, everything in the body is background blood vessels, except for one place, the eye. So we said, maybe we could develop an assay where we could take a tumor and put it in the eye, and people had actually shown that certain types of tumors will, over about a 2 or 3 month period, cause blood vessels to grow. I'll show you pictures in a little bit, to the tumor. And we said, well, if we could have a polymer that we could put right next to it, that A, would not cause damage to the eye, and B, could release any molecule in cartilage. And a lot of these molecules in cartilage were fairly large, proteins, peptides, proteoglycans. If we could do that, maybe we'd have a bioassay. But that was very challenging. No polymer like this had ever been developed. And in fact, if you look in the literature, in 1970s, you often see quotes like this describing this young field. I'll just read this to you. It says, the agent to be released is a small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example, proteins, could be released because of their extremely small permeation rates through polymers. Now, really, the only thing that I had going for me is I just didn't know that. So I went ahead and I actually experimented almost kind of Edisonian-like. I actually found in a couple-year period over 200 different methods to get this to not work. But eventually, I was able to make little microspheres. Here's a picture of one. Here's one cut in half. Now actually we make nanospheres, all kinds of things like this, but this was the earliest demonstration. And we showed in Nature the fact that you could release molecules of virtually any size for over 100 days. Later on, we even showed, using some engineering approaches, how to get constant release rates. Now, when I started this work, you know, I had never given a major talk before. But in 1976, I got asked for the first time to give a talk at a big scientific meeting in Midland, Michigan, where Dow and Dow Corning is. In fact, the only talk I'd ever given before this was in 8th grade. And that didn't go real well. In 8th grade, I remember I had to give a minute-and-a-half talk, and so I stopped work. You know, I spent like 2 nights reciting this minute-and-a-half speech, probably several hundred times in front of my parents' mirror. And then I got up in front of my 8th grade class the next day, and I recited it. And for the first minute and 2 seconds, I did all right. And then all of a sudden, I could not remember the next word, and I just froze. I stood there for another minute saying absolutely nothing until my 8th grade teacher told me to sit down and gave me a not particularly good grade. I think it was an F, actually. So now, many years later, I have to give this talk in Midland to a very distinguished group of chemists and engineers. So I practiced this talk for 2 weeks, this 20-minute talk, and finally the day came and I had to give this talk, and I felt at the end of it I did all right. I didn't forget that much what I was going to say. I didn't stammer too much. So I thought when I was done with this talk that all these, you know, famous engineers and chemists, older ones, being nice people, would want to encourage me, this young guy. And when I got done and stepped off the podium, a whole bunch of them came up to me and they said, "We don't believe anything you just said." They said it's just impossible for these large molecules to get through the polymers. It wasn't until another 3 years later, as often happens in science, lots of people started reproducing what we did. And so then the question shifted. It had to happen, but how could it happen? Well, to understand that, what I began doing is I had a graduate student, Rajan Bawa, and we'd cut thin sections with a cryomicrotome through these polymers to visualize what they look like. So here you see a 5-micron thin section of one of the polymers, ethylene vinyl acetate, and a molecule 300 molecular weight or greater cannot diffuse from one side to the other. But if you put a drug in it, like I'm going to show you a red protein now, and if you put a drug in it, what you see, this is before any release has taken place, is a phase separation. The polymer is here and the drug is here. If you release this for a year, and then you cut a thin section, in sharp contrast to what you saw there, you see where the drug was are pores. And these pores are large enough so that molecules, even millions of molecular weight, can get through. But if you do serial sectioning or scanning electron microscopy, what you see is these pores are interconnected, they have got tight constrictions between them, and they're incredibly winding and tortuous, so it takes a really long time for the molecules to get through them. Myself, actually, I don't know if any of you have ever been to Boston, but I always explain it to people as like driving a car through Boston. I guess a few of you have. Boston is very, so Boston has what we call in chemical engineering very high tortuosity. This does too. And by regulating that tortuosity, you can make these systems last anywhere from days to years, even tiny ones, or any time in between. Well, now since we've done this, we could go back and solve that angiogenesis problem. So here's the assay that I mentioned. Now we had all those 50 fractions. We could put them in these polymers. We did over 1,000 rabbits to 2,000 eyes to look at this, and almost everything we tested didn't work. I kind of look at those as the controls. And they really are in a way, right? I mean, they show that most things don't work. But we found one fraction that did work because of the work that all these people did. Now you see all kinds of angiogenesis inhibitors in clinical use. Probably about 20 million people have been treated by angiogenesis inhibitors. Avastin, produced by Genentech, is probably the #1 or 2 largest selling biotechnology drugs in history. And it's not just for treating all different kinds of cancers, which I'm showing you here. Actually, there's other diseases that these are used for. Probably the leading cause of blindness in this country is a disease called macular degeneration. Before this, the only way to treat it was with lasers. In other words, there were not drugs that existed. And lasers didn't work that well. Now they actually have drugs like Lucentis and others that you can inject, and they can actually even reverse macular degeneration. So we're very pleased that even though this took a long time, it's had a significant impact. At the same time, I thought that not only might we be able to use this as a tool to understand and actually identify angiogenesis inhibitors, but also they should be useful in their own right for molecules that, and drugs that would be tough to take because they're peptides and are destroyed quickly, or because patient compliance is poor. So as an example, Larry Brown in our lab, he did his PhD thesis on insulin, just as a proof of principle. He showed you could take an aspirin-sized pellet, put it underneath the skin of a rat, and you could lower the blood sugar of a diabetic rat for over 100 days. So we worked on these for a long time, and I really wanted people to use this, and actually nobody did. And what I learned is if we want to have people use it, you sort of have to do this yourself. So we filed patents on this stuff. We licensed them to companies, and I got involved in starting a lot of companies myself. And what I find is if you're not your own champion, nobody else is going to be. And through that work, what we see is now many of these on the market. And I'm just showing you a few pictures. But here, through the work, again, of different companies, a number of which actually all of which I've been involved in either as an advisor or as a founder, you see different examples. So for example, if somebody has advanced prostate cancer or endometriosis, there's a very good drug, a peptide, luteinizing hormone, releasing hormone analogs, but it's big, 1,200 molecular weight. You can't swallow it, it won't get absorbed. If you take it nasally or other routes, also you get almost nothing there. If you inject it, it's destroyed almost immediately. But when you put it in these little microspheres, initially the company that made it last for a month. Now it actually is injected once every 4 months. It's treated about 6 million people. Here's another one. I don't know how many of you have seen the movie A Beautiful Mind with Russell Crowe. Anybody saw it? Many of you. Do you remember what disease he had in that movie? Schizophrenia. Do you remember? So he saw people that weren't there. Do you remember how he was treated medically? Electroshock. But today, he'd probably get this. Johnson & Johnson makes a drug called Risperdal, and Alkermes, a company based on our work, developed little microspheres that you could put it in and release it every couple weeks. Again, this has been used to treat about 5 million patients. And there's all kinds of others. This is a once-a-month treatment for alcoholism. This is just approved less than a year ago as a once-a-week treatment for type 2 diabetes. All of these are now being used on tens of millions of people every year. But you always like to think, what's the next thing that you can do in academics that can really change the world? And again, one day in the 90s, I was watching a television show on PBS, and they were talking about how microchips in the computer industry. And when I saw it, I thought to myself, boy, wouldn't this be a great way to make a drug delivery system? Now, again, if you spent over 30 years of your life working on drug delivery systems, you might think that about any TV show. But when I saw it, I talked to Michael Sema. He's my floor mate of mine. He's an expert in ceramics. And we said, well, could we make a drug delivery microchip? And so what we did is we built little wells in these chips. It all is made using formulation procedures in the computer industry, microfabrication and nanofabrication. And you could put different drugs in. You could literally have a pharmacy and a chip. You could cover them with an inert metal like gold or platinum. Anyhow, we made these. I had a very good graduate student named John Santini, and he made some of these for his PhD thesis. Here is a United States dime. Here's a top view of one of them. It's all done by photolithography and other approaches, and you can see all the little wells. Here's one side, here's the other. This, by the way, is a short flat chip. They can be made injectable. You can make them almost any size and shape, but this is just an example. Let me just show you how they work. Here's a well. The drug is actually hermetically sealed. It's underneath the metal like the gold. It'll stay like that for years, except if you just, by remote control, even with something like this, by radio frequency, same way open up a garage door, what you can do is open up the well, and here's an example of that. It's done in nanoseconds. It just comes off, and when it comes off, whatever's underneath it can come out. We published in Nature a number of years ago, in 1999, the fact that you could put different amounts of drug in different wells and release them. We also did more like a pharmacy on the chip idea, releasing different drugs at different times. John, my student, started a company on this, microchips. And over the years, we worked with him and his team to move this from in vitro to animals and in the past year to human beings. And let me tell you about the human trials. So what we've done, and this might almost sound like space-age medicine, but it really happens, and it'll happen more. What we did is the chips, they're actually implanted in the patient. I'll go over that. But you can communicate with them by a special frequency called the Medical Implant Communication Service Band. It's approved, actually, by the FCC and the FDA. And sometimes people ask about tampering, or you might want to change something. Well, there's actually a special computer code. Let's say if you wanted to change the pattern, there's a special computer code you can put in if you wanted to change something. That only could be known to the key people. Also, there's a bidirectional communications link between the chip. We actually, as I'll go over, imprint an antenna on the back of the chip. It's just imprinted, and you can communicate like with a cell phone or a BlackBerry or whatever you want. And that tells you everything that happened, including whether you took the drug or not. I have to admit, as I've gotten older, that becomes more and more important because I keep forgetting, what I've done. And anybody who's getting older, I think that happens to. Let me tell you about the trial. So what we did is this was done in Denmark. We did 8 patients. The disease we picked was osteoporosis. We picked it for a number of reasons. One is we thought we could make an impact. The drug that the women take in this case is, again, a fairly large peptide, parathyroid hormone. Problem is 75% of the time, they don't do it. I mean, who wants to keep injecting yourself every day, especially maybe if you're 70? Also, we felt that this is a good test, right? This is a fairly large drug. Most of the time, people tell us this won't work. We always get that, and you'll hear more about this as I talk. And they'll say because you'll get encapsulation, a molecule that size can't get through. Also, it's a fairly potent drug. And finally, it's not a drug. Like, see, I mentioned before we developed these microspheres that could deliver drug continuously. That would actually be absolutely the wrong thing to do here. That would make the osteoporosis worse. It would stimulate the osteoclast. You need to get what's called pulsatile delivery to make this drug effective. And when we did this, it was done like in a tiny office procedure, 20 minutes. In fact, every woman's survey, we published this in Science Translational Medicine last year, every woman's survey say they prefer this to the shots. In fact, the drug comes out when the women are sleeping. It's all done by remote control. And you got the same pharmacokinetics and actually less variability in the injections. And these are the 3 measures of osteoporosis, same as daily injections. If you look at some of the data, this is just, it was in for about 4 to 6 months. And what you see on this graph is the release rates at different times. This is day 60, day 68, day 84, day 92. They're much less variability than if you injected it. And if you look at the encapsulation, it's not nothing. There's a little bit. This is still about 1/50th of a pacemaker. But and here you actually see the chip. They're actually going to get a lot smaller over time. But here you see the chip, and inside it, it's still a tiny little thing. There's a computer program, electrical components, power source, and in the very back, there's the imprint of the antenna. And just to show you where we're going from this, I mean, there's a whole range of things that this suggests. One, we're going to make 2-year devices and ultimately try to move this as a really practical system. 2, just as one program, Bill Gates actually came to visit me about 8 months ago, and you may know his wife is interested in Melinda about birth control for the third world, that the women could control themselves. So we're actually making a 16-year device. They're actually funding it, where you could the woman could turn it on and off with a cell phone, things like that. Maybe most significantly, what you could do is you could also put biosensors on these chips. So you could actually detect signals in the body on different diseases and then tell the chip how much you want to deliver in return. So there's all kinds of things this suggests. Now, so far what I've done is I've talked to you about how we could use engineering approaches to take materials that already exist and make them do things they never did before. But I want to talk a little bit about the materials themselves. So I mentioned to you I worked in a hospital, and I'm working on materials. I was curious. How do materials find their way into medicine? Was it by engineers or chemists? Well, as I looked into this, I found that was almost never true. Almost always a material found its way into medicine because a medical doctor wanted to urgently solve a medical problem. And they would go to their house and find some object that kind of resembled the organ or tissue they were trying to fix. So in the case of the artificial heart, which has its roots here, what happened is there were people, some of the work happened at NIH, and from a material standpoint, they wanted something that had a good flex life, like a heart. And they said, well, what object has a good flex life? And they said, a lady's girdle. And that's made out of polyurethane. That's what the artificial heart was made out of. when it started in 1967, still what it's made out of today. And one of the biggest impediments in the artificial heart has been when blood hits the surface of the artificial heart, sometimes it forms a clot, and that clot could go to the patient's brain, they could get a stroke, and they might die. But if you think about it, something that's designed to be a lady's girdle might not be the optimal blood contacting material. And this problem pervades all of medicine. Dialysis tubing, a sausage casing. Vascular graft was a surgeon in Texas going to a clothes store to see what he could sew well with. Breast implants, one was a lubricant, another a mattress stuffing. You know, you can probably think of the logic. So what we thought about was, why can't we use chemical engineering design principles to address problems like this? So we picked an example. When we started, the only material that was synthetic and degradable and FDA approved were the sutures. They showed bulk erosion, meaning that you start out like this, but over time they get spongy and fall apart. So if you uniformly distribute a drug throughout this, you'll get bursts of drug coming out, which could be okay for some drugs, but if you had insulin or anti-cancer drug, that might be very bad. You could dump it all out. So from an engineering standpoint, we said, well, we really want this, surface erosion, like the way an onion peels or soap dissolves. How could you do this? So what we said is, well, the way we're going to do this is we're going to ask different design questions. The first question is, what should cause the degradation? Should it be an enzyme? Should it be water or something else? Well, we said everybody in this room probably has different enzyme levels, but you all have excess water. So to make it reproducible, let's build into this material, which we're going to make from scratch, the ability to be degraded by water. But if you want, then we go to the next question. If you want surface erosion, what else do we need? Well, we need the building blocks of this material, of the polymer, to be hydrophobic, to keep water out. Otherwise, you'll get bulk erosion. And then the next question is, if you do that, how will you still make the polymer dissolve at a reasonable rate? So we said, well, we need chemical bonds that when water hits it are going to break fast enough. We calculated you want to go to the anhydride bond and suggested polyanhydrides. And then we'd get to the specific chemistry. We said, well, we want to have polymers that are safe in the body. And here I contacted Michael Marlettis. I was a chemist at M.I.T., also knew a lot about toxicology. Mike's now head of the Scripps. And we came up with this polymer that we drew on the blackboard. And basically, this is a copolymer. Here's one part, here's another. We synthesized these. And what was exciting is not only did this show close to surface erosion, but by changing the ratio of those two units from 0% of the one all the way up to 79%, you can make these last anywhere from 2 weeks to many years or any time in between. So you could dial in the polymer ratio and make these last for almost any length of time you wanted. So one of the things that I've wanted to do, as you can see, is I want to make sure that when we do something that has an impact on patients. In 1985, Henry Brigham, just starting his career in neurosurgery at Johns Hopkins, came to see me. By the way, Henry now is chief of neurosurgery at Hopkins, and I'll tell you a little bit about what he did. So the disease that he wanted to come up with a better way to treat was glioblastoma multiforme. Regardless of how it was treated at that time, and still pretty much today, the mean lifespan is less than a year. The only drug that was approved at that time for use was this one, BCNU. It's a terrible drug. Normally it's given intravenously and it causes terrible side effects because it goes to the liver, kidney, and spleen. So what Henry asked me, he said, could we do something like this? Could you develop a polymer where you could line the surgical cavity, like he's going to operate in the patient anyhow, take as much of the tumor out as he could, but could he leave behind a polymer that would locally deliver this drug by putting in little wafers? Now normally the drug only lasts for 12 minutes. But what they wanted was a polymer that would degrade so it wouldn't accumulate in the brain, that had the surface degrading property so it wouldn't dump this really bad drug, and they wanted it from studies they'd done at Hopkins to last for about 4 weeks. Well, we could do all that with this polymer, and we did. Polymer protects from degradation, but maybe the most important thing is this paradigm, local delivery, high concentrations in the brain where you want it to be, and low concentrations in the rest of the body where it causes harm. So one of the things, of course, that I have to do and that every professor here has to do is if you get an idea like this, like a new polymer, and you have to raise money for it. You have to write grants. So that's what I did. I wrote grants to the NIH, and then they had study sections which are composed of professors from other universities reviewing what we did. I actually saved the reviews. They're not very good. So I entitle this slide, This Approach Will Not Work Because. In 1981, when we first proposed this, the chemists looked at it and they said the polymers could never be synthesized. But I had a very good graduate student at the time named Howie Rosen. Howie later rose to be president of the Alza Corporation, a $12 billion corporation that was ultimately bought by Johnson & Johnson. And Howie's a member of the National Academy of Engineering, and he, for his thesis at MIT with us, synthesized it. We sent it back. The reviewer said, well, it's still not going to work. These polymers have anhydride bonds in them. They'll react with whatever you put in. But I had another couple postdocs, Cam Leung, who's a James Duke professor at Duke University. In fact, it's just going to be announced tomorrow that he's now elected to the National Academy of Engineering. And Bob Linhart, who's Constellation professor at RPI. And they showed that actually there's no reaction. Sent it back again. And the reviewer said, well, we still shouldn't fund it because these polymers are fragile. They're low molecular weight. They're going to break in the body. But I had another couple postdocs, Avi Dome, who became chairman of medicinal chemistry at Hebrew University, and Edith Mathiowicz, who's a full professor of bioengineering at Brown University. And they made very high molecular weight polymers, so using the right catalysts and so forth, and polymers sure didn't break. Sent it back again. And the reviewer said, well, okay, but you did all this, but new materials are certainly going to be toxic in patients. I had another graduate student, Cato Lorenzen. Cato later rose to become Dean of Medicine at the University of Connecticut, member of the National Academy of Engineering and the Institute of Medicine and the National Academy of Sciences. And he showed actually what Marletta had said, that actually now doing studies in all kinds of animals, they were safe. Anyhow, this kept going on until 1996 when the FDA approved it. It was the first time in over 20 years that the FDA approved a new treatment for brain cancer, and the first time they ever approved this idea of polymer-based local chemotherapy. You can probably tell from the way I'm speaking that I'm very proud of how well our graduate students and postdocs have done. They became heads of major departments, heads of major corporations, 3 are in National Academy of Engineering, and they've been enormously successful. The reviewers at the other universities, they haven't done too well. Now, I want to show you what this looks like, but if anybody's squeamish and doesn't like the sight of blood, you shouldn't look. But I'm going to show you this is a human brain, and here's the wafer put in. Usually they put 5 or 6 in, and then they close up the brain. You know, it's very hard to get good advice when you give a talk, but about 12 years ago I was lecturing at this at MIT, and my wife came to the talk. She's a neuroscientist, and I asked her at the end, I said, what did you think of it? And so she said, well, the talk, Bob, she said, your talk was all right. That, by the way, is extremely high praise. But she said, I don't know if you realize, but that you had those 2 bloody slides on for over 10 minutes, and all those poor engineers were turning green and looking at the floor. So ever after that, I did what I did today. I gave a warning. I told everybody, you know, and I showed it really quickly and stuff like that. So just to tell you the sequel to that was that 2 years later, I got asked to give this dinner speech. But this speech was to a group of neurosurgeons. And I did the same thing. And at the end of the talk, a lot of the neurosurgeons came up to me and they said, you know those 2 bloody slides that you showed? And I said, yes. They said, those were fine. They said, you could, no problem. They said, but those chemical formulas. You know, to put those on for a neurosurgeon, I hope there are none here, but they said that's really bad. Anyhow, just to show you some of the data, this was now the, so it did get approved, and originally for recurrent glioblastoma, now for both primary and recurrent. This is for primary. And you see that it does increase survival. You get about, it's not a cure, but you get about a 5-fold lengthening of survival for patients with this. And it really still used 17 years after, significantly still used 17 years after its initial approval. But it also began to create a new paradigm for drug delivery. But before I show that, I thought I would just make a side note. So, you know, there's all kinds of things we do in the lab where you feel, you know, good things have happened. But one day, I remember it must have been about 11 or 12 years ago, I got a call from, I don't know how many different people in my lab, as well as my sister, who's not a scientist at all. And they said, Bob, do you ever watch the TV show ER? And I said, no, I never watch it. They said, well, they talked about your polymer last night. So I actually got you this clip that I'm going to show you. And I should give you an introduction because maybe you haven't watched ER either, but it's actually a pretty good clip. But ER was a TV show, it was actually #1 ranked TV show for a long time. And for a while they had this guy, Mark, he was played by Anthony Edwards, and he was a doctor, but sadly on the show, it's all fiction, he got brain cancer. And what happens is week after week he goes to different cancer specialists and they said, you're going to be off the show in the next week or two. And, you know, it wasn't looking good for him. So finally he goes to one last doctor. And I'm going to show you that clip. I should point out that if you have never seen Anthony Edwards in this, how many of you saw the movie Top Gun with Tom Cruise? So he played Tom Cruise's partner in that movie. He didn't do too well there either. So he died in that movie too in a crash. But anyhow, let me just show you the clip, and if we could, I'll put it on, if I can. Anyhow, so he actually ended up living another 2 or 3 years on the show, and then he did die, which is, and that's unfortunately, it was still a long ways to go. But it does create a new paradigm for drug delivery, and that paradigm has been now used more and more. These were approved in 1996. It's also been used, actually another one of my graduate students, Elazar Edelman, who's now a well-known cardiologist, as well as many companies, Johnson & Johnson, Boston Scientific, and others use the same principles, even our patents, to locally deliver drugs from stents. So you may have heard of drug-eluting stents. It's really the same idea. You take Taxol, which is another anti-cancer, anti-proliferative drug, and if somebody has a stent put in to open up the blood vessel, what happens is, unfortunately, 50% of the time, you get a problem called restenosis, where the smooth muscle cells proliferate. This stops that from happening, and these, like I say, have been used on many millions of patients. What I'm going to do is just going to go to this, talk a little bit, end the talk on tissue engineering, which is what Lon started with. So the other thing that we've done a lot on is people, we don't only want to deliver drugs, but we want to deliver cells. This child is a baby who was dying of liver failure. He was a patient of Jay Vacanti, my main collaborator. Jay's surgeon. He's head of pediatric surgery at Mass General Hospital. And I remember in 1983, Jay said, there's got to be a better way than transplants. Children like this die all the time. Could we come up with a way to take the patient's own cells? This was way before people even talked about stem cells. And could we take the cells, and could we basically use the patient's own cells to form tissues or organs? So the idea we had is, what if we took ways to dissociate any cell type, like this is bone, cartilage, whatever, dissociate them. If you inject those cells into a patient regularly, what happens is that not much. But cells are smart, so if you take these same cells and put them close enough together, they're actually smart enough to reform structures. For example, people at Berkeley have shown you can take mammary epithelial cells, put them close enough together, they'll actually make acini and make milk. So we thought, why can't we make a polymer scaffold that is in 3 dimensions, whatever shape you want, and put the cells on it, and grow it, and the cells could reform the structure. One of the keys to this, which I won't spend too much time on, is having the right bioreactor, because it's not regular tissue culture conditions often don't work. But so actually 2 of the big keys are figuring out the right polymer. In fact, Lon did a great job on that in his PhD thesis with us, and the right bioreactor, and we've had many people work on those. And then the idea is that you could form these different tissues. So I'm just going to give you a few examples to end the talk. First, just to show you some pictures. This is a 3-dimensional scaffold of one of the biodegradable polymers that we use. This is a lactic glycolic acid copolymer with liver cells. But you can make these into any shape. And so Prasad Shastri, when he was with us, he now runs an institute in Germany, basically worked out CAD-CAM techniques, computer-aided design, where he could make these into any shape. So here he actually made a nose. But this nose is 98% porous. You could put cells on it. And this just gives you an idea what you can do. This is like a regular nose. But if somebody wanted to, they could make an upturned nose, right? You could just cut a little bit of this off. If somebody wanted to, they could actually make a hook nose. I mean, I don't know if anybody would want that, but you could do it. And then you could take their own cells, I'll give you an example soon, and make a nose. And even histochemically, it looks like regular cartilage. But it turns out that this cartilage does not yet have the right mechanical properties. Still lots of challenges. Doesn't have the right mechanical properties. But even at this stage, so you couldn't, like if some of you were thinking, could you help a runner? We can't yet. Someday we will. But you can help a lot of people with different issues, and you can make new body parts for people that have problems. And so just to pick an example, we're working, the Army actually came to see Jay and myself, and Linda Griffith, one of my former postdocs, and they have patients come back from Iraq and Afghanistan without ears and without other body parts. Could we help them? So what Linda did is actually make a scaffold in the form of a human ear. Here's a high-powered scanning electron micrograph of it. You see the cells and the matrix. And this has not yet been put on patients. It has been put on rabbits. We haven't put the ear on patients. I hope it will be done in a year or two. But there are a number of cases where we've been able to help patients. This is one of Jay's patients. At the time, he was a 12-year-old boy. But look at him closely. He's got no chest covering his heart. Like other 12-year-old boys, he likes to play baseball. But you could imagine if he ever got hit in the heart with a baseball, he could die. So Jay operated on him. We made a polymer scaffold, new chest, took his own cells, and he's doing fine now. He's actually 6 feet tall. Second example, and this was really from licenses from us, is you could make new skin for burn. This is actually FDA approved for burn victims and patients with diabetic skin ulcers. Let me just show you an example of that. Here's a 2-year-old boy, very, very badly burned. Here's the product. These are neonatal skin fibroblasts on a polymer sheet. You can put it in the child at the time of injury. If you come back 3 weeks later, he's doing better. If you come back 6 months later, he's pretty much healed. And these have now been approved for patients with diabetic skin ulcers and patients with burns. Last example that I want to give you is, and this is still early, is people that are paralyzed. Could you help spinal cord repair? Erin Levick, one of our graduate students, actually made a polymer scaffold. She's now professor at Case Western Reserve, and actually built on some of the work that Lon did. One of the polymers she used was developed by Lon and Denise Barrera, and she incorporated that into a scaffold to change the surface chemistry of the polymer and made a scaffold that has an outer part that can help provide axonal guidance, and an inner part which you can put neuronal stem cells in. And what I'm going to show you to end the talk is studies that first she and Ted Tang did, who was a neurosurgeon, on paralyzed rats. And what was done is we took 50 rats and we caused an injury to the spinal cord, and within 24 hours we did 12 rats that were sham operated, 12 that had cells by themselves, 12 with polymer by itself, and 12 with polymers and cells. So anyhow, the hope though is to try to do some version of this in humans even within the next year. So what I've tried to go over today is to give you an overview of what we've done actually now for the last almost 40 years. When we started, I think the whole area of bioengineering and biomaterials was in a stage where people were using off-the-shelf material, and people really didn't believe in ideas like angiogenesis or drug delivery, and tissue engineering was not even anybody's dream. There's a tremendous distance to go, but it's my hope that this is just the tip of the iceberg, and that these kinds of ideas that we've gone over today are just the start of ways that people in this audience and our students and people all over the world will try to create new kinds of products and discoveries that can relieve suffering and prolong life. Thank you very, very much for inviting me here today.